by now.
Yeah.
So the title is from carbon to complex organic molecules, and I will tell you in a moment what these are. And this presentation is within the topic of prebiotic chemistry and the carbon cycle of the academy. So first I will introduce the astrophysical context of ice and organics in space. Second, I will talk about the laboratory work we do to simulate the processing of ices and the synthesis of organic species in the ice. And finally, I will talk about evidence for the presence of these these complex organic molecules in space. So I always start with this view graph, where you can see the cycle of matter in the universe, which is also the connection between astrochemistry and the new science of astrobiology, where you can start anywhere you want in this cycle. So allow me to start with diffuse interstellar medium. And when the density of these clouds becomes larger, we talk about dense interstellar clouds. In these clouds, we see the formation of new stars, which can be accompanied by a planetary system. And at the end of their stellar life, much of the processed material is delivered back to the interstellar medium. And so these ashes of the star are available for the formation of the second and third generation of stars. So the connection with astrobiology is that in this cycle, there is formation of organics, and these organics are delivered to the primitive Earth by comets and asteroids and smaller particles. So this organic material is probably related to the origin of life on Earth. So this is an exhaustive list of molecules that have been detected in the interstellar and circumstellar medium. I will talk about some of them in this presentation. It's about 300, almost 300 molecules have been detected so far, and every year a few of them are detected. So, well, let's start with one of these dense interstellar clouds. Barnard 68 is a prototype of these clouds, where the density of gas and dust is sufficient to mask the light of the stars in the background. And all we see is that in the outskirts of the star, we see some reddened, of the cloud, we see some reddened stars. And this reddening is due to the presence of dust that is shifting the light of the stars toward the red, because the scattering of shorter wavelengths, let's say blue, is more efficient. So what we get to see is the red component of the background stars. Another famous example of dust scattering is the Pleiades. We took in an amateur group of astronomers in the Guadarrama Mountains here near Madrid. We took during the weekend this beautiful picture of the Pleiades, where you can see the effect of dust very clearly. So this is the dust between the stars and us that is creating this beautiful light scattering. And to do scientific work, we need to do spectroscopy. So images are not enough. So we use infrared spectroscopy to see the vibrations of the different molecules in the infrared. So this is shown here schematically. You can see the symmetric and anti-symmetric stretching of the water molecule and the bending. And thanks to these modes, we can detect the presence of molecules in ices in space, as I will show. So different telescopes on Earth and also airborne, the latest one is the James Webb, have detected these bands in the infrared. In order to do that, we need to have a star that is surrounded by hot dust. This hot dust is acting as the source of infrared light. And then what we need is to have a colder region, so that is the dense clouds, with dust covered by ice mantles. The absorption of this infrared emission is superposed on the baseline, and that's how we get to know what the composition of the dust and the ice is. So you can see that here in a recent paper from the James Webb archive, where you see the presence of the usual suspects that compose the ice, and also the evidence for the dust itself, which is generally made of silicates. Among other species, we see OCS containing sulfur, and this was the topic of one of our colleagues, Asunción, recently. So we composed this scheme of dust covered by ice mantle, thanks to these infrared observations. The ice mantle is formed by accretion of molecules from the gas phase. Some of these molecules require the dust surface to form, like hydrogen or water. So this dust plays a very important role for the formation of key molecules. And furthermore, we are interested in the processes going on in dust. There is a good number of processes going on, as you can see depicted here. So after the accretion of the ice mantle, you can have shocks in the cloud. You have different types of radiation, UV, also cosmic rays, warm up in the, if we talk about hot cores and corinos. And some of these molecules are ejected back to the gas phase. And so we are able to detect them also in the gas. So the end product of this irradiation, based on our lab work that I will describe, is the formation of COMS, these complex organic molecules. And the environments where you can talk about ice processing are interstellar ices, as I said, also circumstellar ones around, let's say, protoplanetary disks. And within our solar system, you could have further radiative processing in comets and icy moons, for instance. So in the case of protoplanetary disks, we are dealing with radiation, forming radicals in the ice. These radicals are mobile, and they can react to form COMS. And finally, by some processing, either warm up when you are close to the protostar, or by radiation, what we know as photodisruption, these molecules are ejected to the gas phase. Furthermore, in the disk, you have dust agglomeration, and forming larger grains helps to preserve these organics. So typical size of dust grains inferred from recent James Webb data is close to 1 micron in size, which is larger than we thought before. And so to wrap up this part of the talk, we can say that we start with dust around young stars, like here in Beta Pictoris. This also occurred in our planetary nebula and in the solar nebula, sorry. And the agglomeration of dust would form comets and also planetesimals. These comets would therefore contain the organics made by ice processing. And finally, this material would be delivered by impacts to the primitive Earth. So Well, in fact, what we observe is that the composition of the carbonaceous material is different from these combs. Typically in carbonaceous chondrites, which are the meteorites with high carbon content, the carbon is in the form of some amorphous carbon material, and only less than 3% in weight of the meteorite is made of what we would call complex molecules. Okay, and this is also the case for the tiny interplanetary dust particles that are collected in the stratosphere. So the composition of most of the carbon bulk is this amorphous carbon, and not the combs. Well, we have some hints for ice processing in the organic globules in meteorites. So this is the case of the Tagish Lake meteorite, but now we know that there are organic globules in many other carbonaceous chondrites. And the interesting thing about these globules is that they are enriched in heavy isotopes, like deuterium, and the size of the walls corresponds to the size of the penetration of UV light. So it could be indicative of formation of organics by ice processing. And therefore, the scenario that we proposed to explain the presence of this amorphous carbon rich in heavy isotopes like deuterium would be a formation in the ice by irradiation. Then the agglomeration into larger particles occurs in the solar nebula, carbonization as well, as the particles get hotter as they approach the protostar, and the silicates become crystalline. So the organic molecules have become this amorphous carbon due to this carbonization. They have lost the nitrogen, and most of the nitrogen and the oxygen. And so the end product would be what we call an IDP nowadays. So if we want to observe the indigenous, the true organic matter with no processing, we need to do observations of combs in the gas phase with radio telescopes like ALMA, with airborne telescopes to detect maybe combs along together with other ice molecules. And we also need asteroidal and cometary missions to detect these combs in situ, like Rosetta for comets or Hayabusa for asteroids. In the second part of the talk, I will focus more on the laboratory synthesis of these organics. So here we have one of these chambers in Madrid that operate under ultra-high vacuum, and they have different techniques to monitor the composition of the dust covered by ice, like infrared spectroscopy, also in the UV, Raman spectroscopy, and then mass spectrometry for the detection of molecules in the gas phase. So we deposit the ice, and then we can irradiate it. As you can see here, after the deposition, we can do the irradiation of the ice, and we can use these different techniques to monitor what is going on in the ice upon irradiation and warm-up. For this, we monitor and we record the temperature of the ice, the pressure in the chamber. We also detect the gas molecules with the mass spectrometer. We can also measure the ice thickness, and the infrared allows to measure the ice itself. And here you can see the tip of the ultraviolet lamp and the deposition window where we grow the ice layer. And we can grow ices of the thickness of one monolayer, so the thickness of one molecule in average. And so we also need the UV lamp with a spectrum that is similar to the one in the interior of these dense clouds. And So it is a hydrogen lamp, and we can also measure the absorption of the molecules we are irradiating in the ultraviolet. We can warm up the ice with different components, mainly water, as in space, and also some carbon molecules, CO, CO2, methanol. And we also introduce ammonia to have nitrogen in there and do some prebiotic chemistry this way. So based on these experiments, we could predict what is the snow line of the different molecules in the in a protoplanetary disk, for instance, and we were also able to predict at which distance from the sun the Rosetta mission would detect water. So the schematic of a typical experiment is the position of the ice, letting gas molecules accrete on the ice, on the cold surface at 10 Kelvin. So that would be the analog of a dust grain. We can irradiate it, and we can monitor the gas with a mass spectrometer, the ice composition with the infrared. We can warm it up to know at which temperatures the different molecules are desorbing. And finally, we can also analyze what remains after all the ice has sublimated, and what is left over is what we call a residue that contains the largest organic molecules that were made by processing of the simple molecules in the ice. So this organic residue that remains at room temperature is also known as yellow stuff, and it's made by radical reactions in the irradiated ice. So, well, first of all, things can become chemically very complicated. So if you take pure ice made of methylamine, you can see that the number of reactions becomes large. And furthermore, you also need to take into account the ice structure, because it also plays a role in the chemistry. And this is what we see here. On left column, you see the ice spectra of, in this case, for acetic acid. And on the right, you see the simulated spectra using density functional theory, which is very useful because it allows to understand what is the assignment of the different bands that we measure, and even related to a nice structure, whether the ice is crystalline or amorphous, for instance. And the third part of this presentation is the evidence for COMS in space. So, well, the simplest case is to look for COMS directly in the ice using infrared observations. For this, we have since recently the James Webb, which is operating very, very well. And here you can see an example where you see the ices toward class 0 protostar, so a very young protostar. And you see the usual suspects there. So water-dominated ice with methanol, CO2, ammonia, methane, and also a zoom on the top inlet in this figure for the presence of COMS. This is very good news in the ice. So you may infer, although it's difficult, I would say, to make a clear assignment for some of these COMS, but this is work in progress. And you can see that there is formic acid probably there, ethanol, and also acetaldehyde. So these would be the COMS detected directly in the ice. Okay. And if we go back to our residue, These are the typical spectra of our residue. And the first thing you notice is that around 3,000 wave numbers, so this is what the astronomers call the 3.4 micron region. You have the carbon-hydrogen bonds, and the structure of this, the profile of this band suggests the presence of molecules ending with OH, with hydroxy groups, what are known as glycols. Okay, so this is just from the spectroscopic point of view, you would say that these molecules are abundant in the residues. Okay, and this is a confirmation of that, because then if you do chromatographic analysis of these residues, you indeed find that many of the components contain a lot of these OH groups, and some of them are very famous ones, like ribose. So ribose is a product of ice irradiation and warm-up, and that is nice because it is of clear astrobiological interest. And furthermore, what we observe in these ices is that if you have sufficient methanol and ammonia to act as a catalyzer, you form a lot of formaldehyde, H2CO, and the formaldehyde will make the polymer, POM, polyoxymethylene, unless you add sufficient water in the ice. So the presence of this POM species is inferred mainly from a band around 1,000 wave numbers in these residues. If you add more water in the ice to make it, I would say, more astrophysically relevant, then you inhibit the formation of these POM polymers. And this is related to a very recent discovery, which I cannot show because it is now under review, where we used the infrared spectra of our organic residues to fit the infrared spectra of nanoglobules in the Ryugu asteroid. So as you know, these samples were retrieved and brought back to Earth by the Hayabusa 2 mission. And so it was very, very good to see that there is a clear resemblance between the composition of the organic molecules in this in Ryugu and our residues. Okay, so let me talk now about another hot topic, which is the synthesis of nucleobases in these irradiated ices. So this is a work by Oba et al., where they found very small amounts of nucleobases, and mainly what they saw most was imidazole, the imidazole molecule. There was no guanine in this experiment, and the question is now, okay, if they are there, even though they are in small amounts, as I said, how do they form? How do these nitrogen heterocycles form? So then I should go back to about 20 years ago, where the presence of the first nitrogen heterocycle in these ice experiments was inferred in the infrared by the two bands that are marked with yellow lines, and that is hexamethylene tetramine. CH2, so there are 6 CH2 groups and 4 nitrogens in the molecule. So spectroscopically, we observe that it should be one of the residue components. And furthermore, when you do the chromatographic analysis, you see many nitrogen heterocycles, And in particular, the most abundant one is this one, that I will refer to as TMT, okay? And the good thing is that this TMT is a precursor of the well-known HMT, the hexamethylene tetramine that you can see here. So, well, the synthesis of HMT is quite complicated, and probably it is not even correct, but this is what we know so far. So you go from TMT to HMT somehow. And what we try to do is to elucidate what is the formation of these nitrogen heterocycles. So instead of using the typical ice composition with water, CO, methanol, ammonia, we decided to start one step ahead using methylamine to form the radical CH2NH, methylamine, because the trimer of this radical would give you the TMT, the precursor of HMT. So we made this irradiation of pure methylamine ice to see whether this synthesis would work. And these are some of the results. You can see that after warm-up of the irradiated methylamine ice, you see the dimers formation, so that is very nice. In particular, the one with the orange frame at the bottom is the dimer that you require to form TMT. So you could see the desorption of some of these dimers during the warm-up of the irradiated ice. And you could also see the desorption close to room temperature, 290 K, of the TMT itself. So a fraction of TMT desorbs close to room temperature. So we also made the trimer, the TMT. And finally, again, in these experiments with methylamine, we could synthesize the typical bands of HMT that are common to the regular ice irradiation experiments. So that was a success in the synthesis of these nitrogen heterocycles. Better known is probably the synthesis of amino acids. So this work is from more than 20 years ago now. We could isolate the different amino acids in the residue and also see whether there was an enantiomeric excess in the amino acids. And since then, there is a good number of follow-up papers on this topic. In fact, during the Rosetta mission, the Rosina instrument also inferred the presence of glycine among the molecules ejected from the comet with mass 75. So, well, it is either glycine or one of the glycine isomers, but in any case, it is an important COM present in comets. Then, in our early work on the synthesis of amino acids, we also made diamino acids. So they contain 2 NH2 groups instead of 1. And that led to criticism of this work because they had not been detected in meteorites. So what we did was to ask for one sample of Murchison meteorite and use the same analytic protocol to search for these diamino acids, and well, they were also there. So that was in favor of a possible synthesis of COMs in these asteroids by ice processing. These diamino acids are components of peptidic nucleic acid, PNA, which according to some authors could be a stable precursor of the more fragile RNA or or DNA. And since this is a protein in nature, the question is what was first, whether the chicken or the egg, the RNA world or the protein world, but this is much beyond my understanding, so I leave it there. Another nice result from the Rosetta mission was the detection of a good number of comps, including acetamide, formamide, methylamine, methyl isocyanate. So it was the first detection of some of these species, and it was the first time that molecules were analyzed in situ in a comet nucleus, directly on sitting on top of the comet nucleus. Also interesting, I think, is that most of these molecules are made by the reaction of two radicals. So in a way analog to the synthesis of comps in our irradiated ices. Well, you may wonder why we observe similar comps when we simulate this ice processing using different sources of radiation, ions, which would act like cosmic rays in space, X-rays, or UV photons. The synthesis of comps tends to be common to these experiments. So if you think of the Lego game, you may think that the starting blocks to make more complex molecules, these are the fragments and radicals you make in the irradiated ice or reactive species like formaldehyde, are not that many. And so, well, they are NH radicals, CO plus or CH radicals. And the warm-up, the way you do the warm-up, faster or slower, is also essential, not just the type of irradiation that you use. So at the end, well, you end up with similar products. Here is an example of well, spectroscopy applied to the detection of comps. So the black dots are the data measured during the Rosetta mission on Comet 67P, and it was fitted with different chemicals. Among them, the one you see in orange was one of our residues. You can say that there is some similarity, but the spectrum of the comet was rather shallow, so I would leave it a little like a doubt. But it is a much better confirmation is delivered by the recent results from JWST organics in these nanoglobules, because there is a clear resemblance between the spectra of the organics in this the Ryugu asteroid and the residues that we produce. But you will need to wait a little bit to know these results. So we are approaching the end of our presentation. So I go back to this delivery of these organics to the primitive Earth. We have some recent examples for this. One of them is the comet Schumacher-Levy 9 impacting on Jupiter in 1994, so it's not so long ago. And also the sun grazers, which are comets that impact on the sun. There are a few of these every year. So these impacts are real, and they are occurring, still occur, although much less than in the past. So this is related, as I said, to the origin of life, presumably, because, well, if you take the mass of 4 Halley comets, that would be enough to account for all the biota on Earth. And during the heavy bombardment by asteroids and comets, that stopped around 3.8 billion years ago. Thousands, millions of these bodies impact the primitive Earth. And we know that life appeared in geological scales right after. So the oldest rocks almost coincide with this 3.8 billion years ago. And in some of those rocks, we see evidence for primitive life. This exogenous delivery did not end. Every day we receive about 100 tons of matter coming from space, most of it in the form of small particles. And this is the end of my presentation. I hope you enjoyed it, and I need to acknowledge my group, my current team, with Cristobal, Hector, Rafael, Bruno, Carlos, a student from Germany, Timon, and myself. This is the current group, and also the previous members of the group, and our international collaborators. Okay, thank you.